My contrast is the powerhouse of the sun. Oh my god! I have a bio test today. I have a chem test this week. I'm sad. I gotta go to school. I gotta learn. Get those co-ops. Get those internships. I gotta keep my GPA. I gotta get a job. I gotta. The complex system of signal pathways in the brain is built upon the process of neurogenesis. A nerve cell can develop into one of three roles in the nervous system. It can become a glial cell, a neurocell, or it can never develop. The neurocell is the foundation for all cognitive and reflexive actions by its characteristic synapse, where neurotransmitters and other hormones are transmitted from the axon of one cell to the dendrite of the other. Glucocorticoids are one of such signals which activate the parasympathetic nervous system in response to stress. The role of neurogenesis is that it creates multiple synapses, creating alternative pathways for cells which act as a buffer in minimizing the effects of stress. If stress continues, though, neurogenesis can actually degrade. In a study comparing mice with and without neurogenesis, it was found that when exposed to stress, mice lacking neurogenesis had increased food avoidance, increased failure in the swim test, a decrease in sucrose preference, and overall increases in anxiety and depressive behaviors. The major conclusion drawn from this is that as neurogenesis decreases, neutralization of stress also does.
A 2006 study explored the link between the elevation of glucocorticoid levels in the brain, lack of sleep, and reduced neurogenesis. It used the small platform method of sleep deprivation to keep mice from obtaining REM sleep. Mice were placed on small, suspended platforms to prevent them from falling asleep, while platforms large enough to cause no disruption were used as controls for factors such as stress. To measure neurogenesis, they used a method in which bromodioxyuridine, or BRDU, a thymine analog which can be incorporated into newly synthesized DNA, is injected into the mice, as described in Toppin 2007. The number of brain cells incorporating BRDU can then be used to track cell proliferation. Greater numbers mean greater cell proliferation and therefore greater neurogenesis. In the experiment, there was no significant difference between mice on large platforms and in the cage control. However, significantly fewer BRDU-labeled cells were present in the small platform mice, indicating that sleep deprivation leads to decreased neurogenesis. In the same study, the authors performed a second experiment to verify that increased brain cortisol levels were responsible for the difference, as they predicted. Half of the mice, shown here as ADX-CORT, had their adrenal glands removed, preventing release of glucocorticoids, while the control mice underwent a so-called sham operation, in which the mice were operated on in the same way, but their adrenal glands were left intact. As predicted, mice with removed adrenal glands showed no difference in neurogenesis between cage control mice and sleep-deprived mice, whereas those that underwent the sham operation showed a significant difference. Some conclusions that can be drawn from these studies are: prolonged sleep deprivation causes decreased cell proliferation and decreased adult neurogenesis. Furthermore, these decreases are due to the elevation of cortisol levels, which is caused by sleep deprivation.
In a 2006 study, 24-hour sleep deprivation was observed using rats undergoing the inverted flower pot technique, being fed every 6 hours. The inverted flower pot technique is one in which a rat is placed on top of an upside-down flower pot surrounded by water. If the rat falls asleep, it falls into the water and therefore wakes up, scrambling back to the top. The cycle continues. A larger platform allowing relaxation while still maintaining a similar wet and isolated environment was used for the controls. After a 24-hour period, it was found that in sleep-deprived rats there was an increase in serotonin turnover in the hippocampus and hypothalamus compared to the control. There was no increase in baseline serotonin values, but rather there was an increase in serotonin turnover in total sleep deprivation of rats across the board. What is serotonin turnover? Turnover is analogous to usage of serotonin. It is the amount that is transmitted across the synapse and resynthesized to replace the amount of serotonin used. This process takes place primarily in neurons. Action potentials going down an axon stimulate vesicles within a neuron to release serotonin into the synaptic cleft. Neurotransmitter receptors on the surface of dendrites readily uptake serotonin across the dendritic membrane. In this way, with an increase in sleep deprivation comes an increase in serotonin production, as well as the amount of serotonin taken up, a process called reuptake. So this goes to show that with short-term sleep deprivation comes an increase in serotonin turnover overall.
In 2011, scientists studied the effects of sleep deprivation on clinically depressed patients. The patients were treated with 36-hour sleep deprivation, and the researchers observed an increase in the production of serotonin. The spike in the serotonin levels acted as an antidepressant for the patients. But is the lack of sleep the cure for depression? If only the human body were that simple. The antidepressant properties of sleep deprivation are short-lived. In periods of long-term sleep deprivation, the spike in serotonin leads to the desensitization of serotonin receptors, which can lead to an increased state of depression.
In conclusion, sleep deprivation is both a psychological and physiological issue. Short-term sleep deprivation acts as an antidepressant because of overall increase of serotonin turnover. Meanwhile, long-term sleep deprivation causes desensitization of serotonin receptors, which can lead to depression, among other mental disorders. It is important to note that sleep deprivation can also lead to psychological depression.
Mr. Sandman.